concepts in embryology for basic sciences curriculum. This is an introduction to the overall basic sciences element of embryology, and then we're going to be following on with limb embryology and of course with the other material that I have online. So the definition of embryology, the definition is the study of the origin and the development of an organism. Prenatal period is before birth, 38 weeks from conception to birth, average fetal age. Gynecological timing has been from LMP, therefore refers to 40 weeks gestational age. So we have the date of conception, which has been at times can be difficult to ascertain. So traditional artificial division, we have the embryonic, which is the period of the first 8 weeks. This is where all the important major organs are formed. So this is a very, very important time because anything not happening right or misfolding at this point will cause organs to not be formed at all or formed in the wrong places. So then we have the fetal period, which is the remaining 30 weeks, and this is where the organs start to grow larger and become more complex. And again, this is also a period in which we can see some abnormalities, which we're going to discuss through the lecture. So the embryonic period, during the first 8 weeks after conception, we have the major embryological events, which are the organs forming from 3 primary germ tissues and the emergence of the basic body plan. So here we can just see from 25 days, 3.5 millimeters up to 36 days, where we have around 10 millimeters up to 56 days, 30 millimeters, where we can really start to see the formation of the human form in more detail. So the fetal period, duration weeks 9 to 38 after conception or until birth. And then major fetal events is organs growing in size and complexity. And here we can just see 18.5 centimeters. This is the size of a 20-week fetus. And you can see this very human-like characteristics at this point. So let's go back to the beginning and look at fertilization to implantation. So as ovulation occurs, the oocyte egg travels up the uterine tube, at which point it may or may not be fertilized with the sperm. So in the case of fertilization, the sperm and the egg meet, and then this would be known as the first stage, which is the zygote. The zygote's fertilized egg. Then as we continue to travel down, we have the 4-cell stage, which is 2 days later. So we can see this is now divided into 4 cells. And then as we continue further down, 3 days later we have a morula, and then 4 days we have an early blastocyst. And we can see the blastocyst cavity, and as we continue on, we follow through into the uterus, and eventually into the cavity of the uterus, we have this implanting blastocyst at around 6 days. And within the blastocyst we can see we have the cavity, and then we have the trophoblast and the inner cell mass. And we're just going to look at how this invaginates into the wall. So ovulation, the egg is released into the peritoneal cavity, travels down the fallopian tube in which fertilization occurs. At conception, in fallopian tube, material and paternal genetic material join to form a new human life, a zygote. Cell division occurs with travel down the tube and into the uterus. Week 1 post-conception, we have the zygote divides repeatedly moving down the tube and uterus cleavage. The daughter cells are called the blastomeres, and the morula, the solid cluster of 12 to 16 of these blastomeres at around 72 hours. Day 4, we have late 60-cell morulas entering the uterus, taking up fluid, becoming a blastocyst. So let's just go through and look at this again. We've seen the early zygote, the full stage, the morula, the early blastocyst, and then down into the implanting blastocyst upon the wall, which we shall look at how this happens. So the blastocyst stage, two distinct types of cells. We have the inner cell mass, which we can see here, forms the embryo. So this inner cell mass is what's going to form the embryo. And then the trophoblast layer of cells surrounding the cavity, this is what's going to form the very important placenta. And it floats for about 3 days, implantation on about day 6 post-conception. Trophoblast erodes the uterine wall. It takes 1 week to complete this erosion process. If inner cell mass of a single blastocyst divides, we're going to have monozygotic, which is identical twins. So let's look at the schematic. Week 2, inner cell mass divides into an epiblast and a hypoblast. The two fluid-filled sacs, the amniotic sac from the epiblast, yolk sac from the hypoblast, we have this bilaminar embryonic disc area of contrast, and this is going to give rise to the whole body. So as we can see, as we continue down day 6, we have the inner cell mass, trophoblast coming down into around day 7, where we have the trophoblast starting to invaginate into the wall of the uterus. We still have the inner cell mass following this, and by day 9 we have a further invagination of the trophoblast, and now we have the additional hypoblast and the epiblast region and the amniotic sac cavity. So as we continue down into day 11, we can see that we have the epiblast and we have the bilaminar embryonic disc, the hypoblast, the yolk sac cavity. So all of this we can see, and we can see this invagination through into the wall of the uterus, and an almost complete transference through at this point. So week 3, there's a few things going on that we need to just be aware of. Okay, this is where we're going to go from the bilaminar to trilaminar disc. There's 3 primary germ layers. All body tissues develop from these. These are the ectoderm, the endoderm and the mesoderm, and we're going to talk about these and look at them in simplistic ways just to have an understanding of what they do, where they go, and essentially how they do all they need to do in the body. So here we can just see a bilaminar embryonic disc, lateral to superior view. We can see the primitive streak, and then we have the hypoblast and the epiblast, and we have the region of the head, the yolk sac, and this is going to be that region of the head, so it's going to be cranial, and then tail caudal, and again we can see bilaminar embryonic disc, right, lateral superior view. Again we have the hypoblast and the epiblast. We can see the head region and the tail, and then just looking from above we can see the primitive streak, the primitive node, and we're going to start to consider how we go from this bilaminar to trilaminar positioning, and then how we start to get the important foldings, which are going to make a lot more sense to when we consider things like body cavities. So here we can just see the epiblast and the hypoblast, the primitive streak, the endoderm, and then eventually we have the ectoderm, the mesoderm, and the definitive endoderm at around 16 days. So formation of the germ layers. So the primitive streak groove on the dorsal surface of the epiblast, graduation, invagination of the epiblast cells. Days 14 to 15 they replace the hypoblast, becoming the endoderm, and then day 16 the mesoderm, a new third layer is formed in between, and then these epiblast cells remaining on the surface ectoderm. So again, let's just have a quick look at these different images. We have the bilaminar embryonic disc, lateral to superior view. We've got the hypoblast, the epiblast, region of the head and the region of the tail. Just from an overview, we can see the head, tail, left and right region, the yolk sac and the cut edge of the amnion. And then from days 14 to 15 we see the primitive streak, the endoderm, and then as we progress through to day 16 we have this mesoderm as well, this definitive endoderm and the mesoderm, which we're going to start to look at. So the 3 germ tissues tissues. These are, as in germinate or not germs, as in germs that one might catch. Early specialization of cells, they are precursors. The ectoderm and endoderm are epithelial tissue, which is going to form sheets of tissues, which we're going to start to look at. Mesoderm is mesenchymal, mesenchyme tissue. Mesenchyme cells are star-shaped and do not attach to one another, and due to this they can migrate freely. The notochord at day 16 to 18 is primitive node epiblast cells invaginate and migrate anteriorly with some endoderm cells, and we have this rod defining the body axis, which is going to be formed. This is going to be the very important site of the future vertebral column. And here we can just see the amniotic sac, the plain section of the head, and we can see the primitive strake and the primitive node. We have the notochord and the amnion, and here we have the embryonic disc, the ectoderm, the mesoderm, and the endoderm. And this is the yolk sac. And here we can just see from the right and from the left, we have the invaginating mesodermal cells between the endoderm and the ectoderm. And this is going to start to make more sense as we start to look at how this all comes in and then ultimately folds in and zipper-likes together. So, neurulation. The notochord signals the overlying ectoderm. Formation begins of the spinal cord and brain. This is known as neurulation. The neural plate to neural groove to neural tube is pinched off in the body. So here we have the tail and the head, left and right side, the primitive streak. Now here, as we look at this cross section, we have the surface ectoderm, the neural plate, the future neural crest, and this is of course the region of the notochord. And here we can see the amnion, the amniotic cavity. We have the neural plate, we have the ectoderm and the mesoderm in the middle, and of course we have the notochord and the endoderm posteriorly, and the yolk sac, which are all going to eventually fuse in. So by day 22 we start to see this neural folding. We have the somite. Here we have the neural fold. This is going to be the tail edge, the cut edge of the amnion. And then if we were to imagine this would what it would look like on the human overall. This is the ultimate design that we're looking to get to. So here we have the neural tube itself, the surface ectoderm, and we have these neural crests and the notochord, and then the surface ectoderm, neural crest, and the neural tube, and we have the somite and the notochord. And we're going to start to look at how this is going to ultimately fuse over into the midline and form the structures that we notice in adults. So closure of the neural tube begins at the end of week 3, complete by the end of week 4. Folic acid is important for this step and extends cranially, eventually the brain, and caudally the spine. The neural crest, lateral ectodermal cells, pulled along and form sensory nerve cells and other important structures. And here we can just see at C20 days we've got the neural fold, the neural groove, the somite covered by the ectoderm, its primitive streak, and it's all beginning to fuse in. Here we have the neural crest, and we can see this region here, the colon, which we're going to talk about in a moment, the neural crests and the somite, the neural fold, and then we've got the intermediate mesoderm, and then we have a lateral plate, splanchnic mesoderm, and a somatic mesoderm as well, which we're going to talk about. So the mesoderm begins to differentiate. We've got the lateral to the notochord, week 3, extends cranially and caudally from head to tail or crown to rump. Divisions of the mesoderm into 3 regions. We've got the somites, 40 pairs of body segments, repeating units like building blocks by the end of week 4. intermediate mesoderm just lateral to the somites. Got the lateral plate which splits to form the colon, the cavity, which we can see here. So the divisions of the mesodermal lateral plate, we've got the somatic mesoderm opposed to the ectoderm, splanchnic mesoderm opposed to the endoderm, colon in between, which will become the serous cavities of the ventral body cavity, peritoneal, pericardial, pleural. So folding begins at week 4, and the main differences between the 3-week embryo and the adult body is that the embryo is still a flat disc. So here we can just see this flat disc of ectoderm, mesoderm and endoderm, this trilaminar embryonic disc. And then what we're going to have is we're going to go from here, we get to our lateral fold, which is going to form this future gut from the digestive tube. You can see here that this is going to have the endoderm being formed on the inside. So as we continue to progress, the 24 days, the embryo protrudes into an amniotic cavity. We see the somites seen through the ectoderm, and the head and the tail are all starting to fold in, and the yolk sac is moving as well. The future anal canal will be in this region that we can see, so we can see the neural tube, the notochord, the primitive gut, and then of course we have the future opening between the mouth and the pharynx. So all of this is starting to happen at this very important time. So day 23, we're continuing with this folding, and then we can see the ectoderm and the somite and the neural tube is forming, the notochord and the endoderm are still continuing round into the midline. The somatic portion of the lateral mesoderm is developing colon, and then the splanchnic portion of the lateral mesoderm. And then we can see that we have, as we continue through, we have the somatic mesoderm and the colon, this actual area here now, from when we look at it before, as we progress through, we have these further walls. We have the region which is going to become the future gut, the future digestive tube, and then this splanchnic mesoderm, which we're going to look at as we progress through. So the cylindrical human body plan is accomplished by day 28. It's about 0.5 centimetre wide. And here we can see the somite, the dermatome, the myotome, and the sclerotome. We have the neural tube, we have the ectoderm, the epidermis, the ectoderm, the gut lining, the endoderm, the limb bud. So we're going to talk about limbs. That's going to be our next lecture on limb budding. And then we're going to have the parietal serosa, the somatic mesoderm, the dermis, the somatic mesoderm, and the peritoneal cavity, the colon. And this would be the smooth and connective muscle of the gut, the splanchnic mesoderm. So if we just look here, this is a simplified cross-section through the abdomen of an adult, essentially the same as above. So you can just see from even at this very early stage at day 28, and then looking at the adult formed system, you can imagine how all of this is eventually going to the neural tube form here, and of course the other areas pushing in midline or anteriorly into the midline. So here we can see the spinal cord and the vertebral column and the kidneys. So this is all going to be the future site of the kidneys, and the inner tube lining of the digestive tube, and the muscles of the digestive tube, and the viscera, and these various visceral layers. So this is important to understand, because when you consider general visceral efferents, general visceral afferents, general somatic efferents, general somatic afferents in relation to the body wall, and you think about the visceral layers, what's visceral, what's parietal within the peritoneum, or the pleura, visceral pleura, parietal pleura, and visceral peritoneum, parietal peritoneum. The understanding of the embryological development really helps with that, and of course it helps with the transition of the veins, arteries and nerves from the posterior aspect round to the midline. So the major derivatives of the embryonic germ layer, let's start with the ectoderm and look at what the ectoderm is going to form. So we have the epiblast, the ectoderm, epidermis, hair, nails, glands of skin, brain and spinal cord, neural crests, sensory nerve cells and some nervous structures, pigment cells, portions of skeleton, blood vessels in the head and neck. Then we have the mesoderm, which is going to give off the notochord, which is going to be the nucleus pulposus of the IVDs, the intervertebral discs. Then we have the somite region of the mesoderm. It's going to be the skeletone, vertebra and ribs, the dermatome, dermis of the dorsal body region, and the myotone, trunk and limb musculature. And then we have the intermediate mesoderm, which is going to form the kidneys and the gonads. Then this lateral plate mesoderm is going to branch off into two. We've got somatic and splanchnic. The somatic, parietal serosa, dermis of the ventral body region, connective tissue of the limbs, bones, joints and ligaments. The splanchnic mesoderm, wall of the digestive and respiratory tract, except for the epithelial lining. Visceral serosa in the heart and the blood vessels. And finally, the endoderm is going to be responsible for this epithelial lining and glands of the digestive and respiratory tracts. So here we can see that 26 days, the embryo, this is when the heart starts pumping, about 4 weeks or 1 month, around half a centimetre size. You can start to see somewhat of a human-like figure starting to appear. We have an ear and pharyngeal arches, the eye, the heart, the upper limb bud, and the tail, the lower limb bud, and so the somite soon gives rise to the myotomes. Then at 3 months fetus, around 6 centimetres, we have a far more human looking configuration at this point. And then later on, late 5th month, we're around 19 centimetres, where we're a lot further developed in relation to structures. So the embryonic period, duration first 8 weeks after conception, major embryological events, organs form from the 3 primary germ tissues, and emergence of the basic body plan. Then by the 8th week, we've got about 2 months, all major organs are in place in at least a rudimentary form. This is why drugs early in pregnancy is so important to avoid, because many of these cause birth defects and the baby's little over. So it can affect the developing fetus in a great deal of many ways. Drug taking, smoking, excessive alcohol can stunt the growth and can also cause retardation of certain aspects of the growth cycle. So again, we can just see the duration, week 9 to 38 after conception or until birth. Got major fetal events, organs have grown in size and complexity. Here we can see around a 20-week fetus, 18.5 centimetres. Then 9 months shown in mother's womb, uterus, preparing for birth. And of course you can see the umbilical cord, the placenta, the amnion, and the wall of the uterus, and the eventual passage in which the child shall pass into the world. So any questions, please just put the comments down below. This is going to be the first basic embryo introduction, and then the next one is going to be on limb bud development, which is going to come up in the next couple of weeks. And then these also link with my series on the nervous system. So please feel free to, as I say, leave me comments or anything you would additionally like covering, material you'd like covering, and also please feel free to access to my free 3D online library. You can use these models, you can download them, and you can also 3D print them as well, so if you should so desire.